if we use an analogy of playing a piano. So playing certain keys in a certain sequence on a piano plays a really nice tune. What a somatic mutation theory is kind of suggesting is that it doesn't matter what keys you play, you will still get the same tune. It doesn't matter what mutations occur, you're still going to get the same growth in exactly the same manner, following the same path.
Mm.
Well, it's very difficult to explain how utter randomness can cause that consistency. But it's not just that, because we find that there's a number of cancers now that develop without any driver mutations at all.
Mm.
So bare minimum in those cancers, something else is driving it.
Welcome to Ultralife Today. Today we have an amazing guest, Mark Lintern. You know, for decades, cancer has been understood primarily as a genetic and cellular malfunction, a story of mutations, signaling errors, and metabolic breakdown. That framework has produced real progress, but it has also left important questions unanswered. Modern tumor sequencing generates far more information than just human DNA. For years, much of that additional genetic material was ignored. But in 2022, researchers went back and asked a simple but profound question. What else is living inside of tumors? What they found was fungal DNA, not randomly, not occasionally, but in structured patterns that varied by cancer type and repeated across countries and laboratories. This discovery doesn't just discard everything that we know about cancer, it expands it. Our guest today is Mark Lintern, author of The Cancer Resolution. Mark brings together these findings with metabolic theory and immune biology into a unified way of understanding cancer, not as a simple accident of broken cells, but as a living biological ecosystem. Today's conversation is about this new perspective and what it reveals, and how it may reshape the future of cancer care. Mark, welcome to the show. How are you doing today?
I'm doing fantastic. Thank you. Thank you for having me on the show. It's a pleasure to be here.
So this conversation today, we really want to dive into kind of this unified model that you've developed. And for listeners that are new to your work, how would you describe cell suppression theory in clear, everyday language?
It could be seen as a meta theory that explains a number of key parts of the cancer puzzle that still remain unexplained, but it sits as an adjunct to the other theories that are currently available and highlights these areas to show how all these different theories fit together and where all these other parts of the disease that remain unexplained can be explained through this process.
I have with me also in the studio my dear friend Kyle Drew. Kyle, you were a part of a whole ecosystem with Know the Cause by Doug Kaufman.
Yeah, Doug Kaufman, author of the Fungus Link series of books and host of Know the Cause. And Mark, when I began reading your work some time ago, I was enamored and I was heartened. And again, I think that so much of what you've brought together represents a huge missing piece of the puzzle. But it's not just one piece, Mark. And this is one of the things I wanted to ask you. You talk in your book, The Cancer Resolution, about the hallmarks of cancer, the 10 agreed upon hallmarks of cancer. And a few of them might be something like uncontrolled growth or immune evasion. And the dominant theory of cancer is the so-called somatic mutation theory.
Wah, wah.
Exactly. And while that's the dominant theory, it really only hits a couple of those hallmarks. Would you mind talking about that a little bit and how the cell suppression theory that you've developed helps to bridge the gap between just the small number of hallmarks versus the greater one?
At its core, though, I just want to say
Yeah.
this. The somatic theory has failed, not just
Oh.
because I don't think it's not a great theory, but all that pharmaceutical science has tried to build as an answer to the somatic defect theory has failed. All of these gene interventions going in that should essentially answer the defect that's going on in cancer, the efficacy less than 2% for most of these interventions.
Yeah, and I'm going to mention something about that. But Mark, talk about the hallmarks of cancer and why somatic theory fails
Yeah,
so much for you.
why does it fail?
Well, I can understand why science has gone down that route, from the 1950s. There's only so much we know at any given moment in time. And it was an exciting time when they understood the structure of DNA. And it was thought to be kind of like the God particle, really, isn't it? The God
Yeah.
part of the cell you can create. If you can manipulate and understand the DNA, you can create whatever you want or solve any particular disease in that sense. But since then, it's been shown that actually probably about 1.2, 1.3% of mutations actually relate to any particular disease in general. That was John Ioannidis, his paper. I forget the date of that paper, but he did an extensive study on that to show that, DNA mutations aren't exactly the direct cause of most disease. In fact, they may add to those diseases. But throughout the decades, I can see why so much investment went into trying to understand this very complex mechanism behind essentially the blueprint of the cell. But we know it's not so much that anymore, because there's a lot of epigenetics that affect everything that's going on. We get into the future, everyone's assuming that this is the underlying mechanism behind the disease. We have lots of investment. So you've essentially got this big juggernaut now going full steam ahead, trying to understand the genetics of cancer. The Human Genome Project came out. That took about 15 years to literally catalog all the DNA genes. So still, we didn't really have enough information. We weren't able to catalog everything that was going on in the cell at any given moment in time. And sure, there were mutations happening. So you can see why people scientists still believed that this was one of the main mechanisms driving cancer. But it wasn't until the Cancer Genome Atlas project, which started around 2003, 2005, where technology had improved so much that they could now catalog the DNA mutations occurring in a broad number of cancers in a relatively short period of time. Rather than 15 years, it was taking 6 months, a year or so. But when they did release that data, they began to realize that the data wasn't showing the kind of results that they were expecting. So they're expecting sequential mutations, like a pattern of mutations that you would have with skin cancer or whatever type of cancer it was. And that would be the same for everyone who had that particular type of skin cancer. It would be a pattern. We could target those specific mutations with drugs. However, in 2013, when a lot of the evidence came about and it was analyzed, a lot of the most of the mutations were random, utterly random. And it didn't really make any sense at that point. So the key point I would highlight here is the reason why it doesn't make much sense is because those 10 hallmarks you were talking about, Kyle, they represent the consistency of the disease. So here we have a prospect where you've got utter randomness, and we're not just talking randomness from one cancer to the next. We're talking about randomness of mutations within different cells of the same tumor.
Right.
Yeah. So it wasn't it wasn't a consistent defect that they were seeing in any particular cancer cell.
And it wasn't just the randomness, but if you think about this on the level of, like, if I use an analogy of playing a piano, so playing certain keys in a certain sequence on a piano plays a really nice tune. What a somatic mutation theory is kind of suggesting is that it doesn't matter what keys you play, you will still get the same tune. It doesn't matter what mutations occur, you're still going to get the same growth in exactly the same manner, following the same path. Well, it's very difficult to explain how utter randomness can cause that consistency. But it's not just that, because we find that there's a number of cancers now that develop without any driver mutations at all. So bare minimum in those cancers, something else is driving it. Even if it would be genetic mutations driving the other forms of the disease, or you still speculate that could be the case, you now have a set of cancers that need to be explained by another mechanism. And that should open the doors to a lot more research into trying to find out what that other mechanism can be. But it seems to have been sidelined. And then if you go down that route and you actually do find that other mechanism, you could then argue, well, how do we apply that mechanism to the other cancers that have got mutations? Because it might apply to them as well. But it just seems like no one's really taken the time to go down that route and look at these other cancers that don't have any mutations. But Then there's recent studies that have shown that the average mutation rate is about 1.7 mutations per cell. And, there aren't enough mutations essentially to drive the disease under this somatic mutation model. And
Right.
then there's mutations in healthy tissue that you don't
Right.
get cancer developing. Then you have the DNA transfer experiments where they essentially took the nucleus taken from tumors, and they placed that into healthy tissue, healthy cells, and they expected to see abnormal growth because that's what you would expect to see.
Right. Yeah.
Yep.
You take the DNA from a cancer cell, put it into a healthy cytoplasm. This is what Seyfried did in his whole group with the metabolic approach to cancer.
He
Right.
did.
And
Great.
what happens is exactly the opposite, right? So the cancer nucleus in the healthy cytoplasm, guess what? Stays healthy. And you take a healthy nucleus, put it into the cancer cytoplasm, and it turns cancerous. So, somatic mutation theory gets thrown out the door.
Well, and Mark, one of the things, and I just want to point this out for folks, on your website, cellsuppression.com, you have this wonderful synopsis paper, the cell suppression theory synopsis. I hope everybody goes to your website and looks at this and then gets a copy of your book. One of the things you were just talking about this.
And where can they get the book? It's Cell Suppression Theory.
Yeah, cellsuppression.com.
Yep.
And then I got mine on Amazon, Mark.
Yeah.
My copy of the book. And you're talking in this synopsis about the shortcomings of the dominant theory. And then you switched over to talking about the 5-year survival rates, the average 5-year survival of various cancers. And you start listing them out. Liver cancer, 5-year survival when you are at stage 4, 5%. Pancreatic, 3%. Stomach, 6%. The highest 5-year survival at stage 4 is non-Hodgkin's lymphoma at about 30%. In other words, by funneling so many resources into the somatic mutation theory, the results are often single-digit survival rates at the 5-year mark at a certain stage of growth.
Let's help our listeners here because some of them might be a little bit confused.
Okay.
Somatic theory of cancer is essentially the predominant theory, which says that defects in genes is what causes cancer. And so, guys, that's when we say the somatic theory of cancer is proving invalid. We're saying that this whole theory about your genes getting damaged, causing cancer, let's throw that theory out with the bathwater because it's proven at its core not to be valid. Now, genes do play a role, but really what we're talking about is a new theory that Mark Lintern has put together in the Cancer Resolution. And let's get back to the question that you're asking there. Just want to make sure people understood, because when we say somatic theory, we understand it.
Right. Genetic mutation being the central
core
cause of cancer, that's what the theory states. And so thank you for summarizing that, because Mark, when I was reading this, you really did a beautiful job of putting the horror story in black and white of, well, how are we doing after kind of using this approach for 75 years? How we doing so far? Are we making progress? And Mark, to me, I'm not sure that the progress has been worth the investment, but what are your thoughts?
Yeah, I agree. And that was one of the big issues with my research. It became clear quite early on when I started my research. I mean, I believed initially that cancer was a genetic disease because that's what we were all taught, right? So I was searching the medical literature to find the mutations that caused my particular cancer when I was diagnosed. And it's only then that you realize that it's not related to much that is out there. There's no pattern.
You mention your own diagnosis. I am assuming that this is what fueled your interest in really going deep and staying down long and coming back to the surface with more answers than questions. So maybe talk a little bit about your own story and what has fueled this research.
Yeah, so I developed cancer when I was 28. It was just a skin cancer, essentially. So I was lucky I managed to get it removed via surgery, but that threw me into researching the disease, basically. And it was quite quickly after that, after about 3 to 4 months of doing research, I came across these other theories, and a number of issues I had already surfaced with the genetic theory, the randomness that I saw in the research. And then I stopped after I'd been given the all clear, essentially. A couple of years later, a good friend of mine, Sam, she developed cervical cancer at the age of 30.
Hmm.
So I began my research again, and that lasted a year. I did a year's deep dive because after the year, unfortunately, she passed away.
Oh,
Hmm.
my.
Goodness.
In that process, I was trying to, point of the research was to help her as much as I could to provide other potential adjuvants she could use to try and help her through chemotherapy and various other things to improve her survival. But unfortunately, she went down the um, um, mainstream treatment route, standard of care, and within, like I say, a year, she passed away. So that provided me with the motivation, really. I didn't want to go through that process again. I didn't want any of my loved ones to experience that. So I thought I've understood quite a bit about the biology of human biology and cancer biology. I'll put what I know down in a little pamphlet or something, so that if anyone I know gets cancer again, I can provide them with that. And that would set them on a path of interest, looking at other things, not just going along with standard of care. I'm not saying that standard of care can't help. It can in some cases, but this would help anyone who developed the disease on my side. And then, of course, because I'd learned so much, I didn't want to give this away. I was motivated. I was angry because of Sam's death. I could already see things that were going wrong. I mean, she was being given Avastin 3 months before she passed away. And I'd already researched Avastin and realized that it had been taken off the market previously for breast cancer because it showed no long-term benefit at all. So that motivated me. I carried on going. I then came across the metabolic theory and a number of other theories. But for me, it became clear that metabolic theory was one of the leading established theories.
Right.
And then one thing led to another. I was writing a book at this point. It wasn't just a pamphlet. It was a book. But then I came across a couple of contentions with the metabolic theory itself. That led me on to some further research. I wasn't even looking at cancer at this point. I was looking at infection-based models, and something struck me in that paper. And they described the Warburg effect in that paper, nothing related to cancer, but they were talking about how infection triggers the Warburg effect. Now, the Warburg effect is what the metabolic theory describes as the underlying mechanism that drives cancer. And that is when the energy system switches from, you could say, the primary energy system or the primary energy system to the backup energy system of glycolysis.
Mm-hmm.
So cancers favor that process. And that's been termed the Warburg effect because Otto Warburg discovered this shift in energy that's abnormal for cancers. But I've basically come up with a completely different explanation for why the Warburg effect could be occurring in cancer. And it's all related to infection.
And when you discovered this, I'm sure that you were thinking, oh gosh, this is kind of outside the mainstream. But what I think in my mind, what helps is the work of, for example, Thomas Seyfried and the metabolic theory. I know that this is not contending with metabolic theory. It's working in conjunction with metabolic theory to give a more rounded, full approach to the concept of how do we get cancer, and by extension, how should we be treating cancer? Am I going kind of balanced with that?
So this is pointing to the question, what is fungus doing in our bodies that's causing this cancer? So you made the link that fungus infections can cause cells to have the Warburg effect being expressed. What is going on to bring it from a cell having the Warburg effect? How is fungus causing the cell to turn into a cancer phenotype?
So it would be best to explain this through Dr. Navier's cell danger response model.
Yeah.
He describes, so if we describe cancer first through the metabolic theory, the notion there is you have these two synergistic energy pathways. There's more energy pathways than that, but these are the main two. So let's call it OXPHOS, goes through mitochondria. That's where glucose and oxygen is converted essentially into energy in the mitochondria, which are the organelles which resemble bacteria.
And it's a very efficient system. One glucose makes, I think, 42 ATP, which is incredible.
Yeah, it is one glucose molecule. But the issue of that is that oxygen is required to produce energy. So if you have an event where you have damage to the cell and you have hypoxia, loss of oxygen, the cell needs to have a backup energy system in order to survive, essentially, for a short period of time until the blood
That's
vessels are.
convenient.
Yeah. So they have this backup energy system called glycolysis. I mean, there's two parts to that, but won't really go into the detail, but just to keep it simple. Glycolysis, that produces lactic acid. And what lactic acid does is it actually stimulates blood vessel growth. So there's a switch over to lactic acid to glycolysis, which is where the cell ferments glucose. So it doesn't need oxygen. So that's beneficial, obviously, for when you lose the oxygen supply. Now, that produces around 2 ATP units of energy for every glucose molecule. So you need at least 18 times, 20 times glucose molecules to provide the same energy output that you would do through mitochondrial oxygen.
Yeah, it's extremely less efficient, but
Yes,
it's the backup, right? So it's there.
absolutely. It's not really designed to be on for a prolonged period of time.
Right.
So that's the difference. That's what happens with cancer. And it's known as the Warburg effect, because what we see in cancer is that oxygen is still being absorbed into the cell. So if oxygen is still being absorbed into the cell, technically, mitochondria should still be using that oxygen to produce energy. The fact that we've got this switch to glycolysis, the backup energy system, even in the presence of oxygen, suggested to Otto Warburg that mitochondria must be damaged. So it's forcing this switch. Now, that's the basic premise. The OXPHOS pathway in mitochondria are dysfunctional to a certain degree that it no longer produces sufficient energy, ATP energy, for the cell to survive. So it is forced to switch over to this backup energy system, corrosive lactic acid producing energy system. Now, the difference with my theory is that when a fungal pathogen is detected or invades the cell, and I'll go through this as Dr. Robert Navia explains it, because he created something called a cell danger response model. And this model explains what happens when the cell experiences danger, a toxin, damage, pathogen, whatever it is. It goes through a 3-step process. CDR1, which is the initial process of alerting the immune system, inflammation, switching over to glycolysis. And the whole point of this is to eliminate the pathogen or the toxin. Stage 2 is where the signaling comes in more intensely regarding repairing the tissue. So you've got to assume that stage 1 has been going on for a short period of time, as say an acute inflammation. You damage the tissue, you eliminate the pathogen, it moves over to step 2. You then need to repair the cell. So you're now drawing in a lot more nutrients so you can repair the cell and the tissue. And then it moves on to step 3, CDR3, which is essentially dampening down all these mechanisms for cell repair, and the tissue returns back to homeostasis. Well, how Dr. Navia describes an infection in the CDR1 phase is that the pathogen is acknowledged by the cell, and it's the mitochondria specifically that are monitoring everything going on. There's so much more than just an energy organelle. They're actually the cell defense mechanism. They monitor many different aspects of cell functionality. And one of them is when pathogens attack. When that happens, the mitochondria intentionally suppress OXPHOS. So they intentionally suppress the pathway that is thought to be damaged in the metabolic theory. So that suppression makes it appear as though mitochondria aren't working and forces a switch in energy to glycolysis. Because now that mitochondria aren't producing ATP, because what they're actually doing is they're taking that oxygen being absorbed into the cell to actually target the pathogen with reactive oxygen species. Also, reactive oxygen species are used for cell signaling. So they're signaling for inflammation to alert the immune system to the issue. So they're repurposing the oxygen. That means that they're no longer creating ATP energy, but they're using it proactively to defend the cell. That forces this switch to glycolysis, because glycolysis is now providing the ATP that allows the mitochondria to do their other job of defending the cell. So that's essentially the shift that occurs, and that offers a new and novel narrative for the reason why the Warburg effect could be occurring in cancer. It's the fungal
Yeah.
pathogen that's driving that process.
Yeah. I think we're going to bring a close to this first segment here. And Kyle, do you want to just frame where we've been and how and where we got to here for the moment?
Well, when we are confronted with cancer, we are hit with one dominant theory when we go the conventional medicine route, and that is something called the somatic mutation theory, which just means that the DNA has mutated and then it's done spontaneously. We don't know any particular reason why, and we have cancer. All of the treatments, most of the treatments that are available to us are reflective of that theory. But what if that theory is wrong? What if there is another approach? And our guest today has been Mark Lintern, who's written The Cancer Resolution. The Cancer Resolution. And there's a question mark behind that. I don't think that you need a question mark, Mark, but that's just me. But the Cancer Resolution, his website is cellsuppression.com, cellsuppression.com. The cell suppression theory is what Mark has developed, and I think that it's got some real steam behind it.
It's got some bones to it.
It does, and I think, Mark, that you've done a tremendous work, and I'm looking forward to our next conversation. But thanks for being with us today, brother.
Yeah, thanks for joining us today at Ultralife Today. Please come back to us next week when we continue our discussion with Mark Lintern.